A warm welcome to you from SGT University. I'm Dr. Mona from the Faculty of Medicine and Health Sciences. Today we'll learn about the determination of blood groups according to the ABO and RH system. Determination of blood groups is important in blood transfusion, in pregnancy in case of RH incompatibility, in paternity disputes, and it has medico-legal value. Also, the blood group is the unique identifying feature of each individual. In order to understand what blood group is, the student must know that either the A antigen, B antigen or RH antigen is present or absent on the surface of the RBC cell membrane and the corresponding antibody is not present in the plasma. For example, if the blood group is A positive, it means that the A antigen and the RH antigen are present on the surface of the RBC cell membrane and anti-B antibody is present in the plasma. Similarly, if the blood group is AB positive, it means that the A antigen, B antigen and RH antigen are present on the surface of the RBC cell membrane and no antibody is present in the plasma. Similarly, if the blood group is O negative, it means that no antigen is present on the surface of the RBC cell membrane and both anti-A and anti-B antibodies are present in the plasma. All the other blood groups can be derived from this basic principle.
The apparatus for this practical consists of a kit containing anti-serum A, anti-serum B and anti-serum RH, normal saline which is 0.9% NaCl, 5 glass slides, a watch glass and dropper.
In order to perform this practical, we need the following apparatus: a kit containing anti-A, anti-B and anti-D antisera. Inside it, this blue bottle contains anti-serum A. It has artificially prepared antibodies against the A antigen. The yellow bottle is anti-serum B. It has artificially prepared antibodies against the B antigen. And the colorless is anti-serum RH. It has artificially prepared antibodies against the RH antigen. Besides this, we also need a bottle of normal saline which is 0.9% NaCl, a watch glass, a dropper, a lancet and a spirit swab. I also have a vial containing anticoagulated blood which I am going to use for demonstration purpose. Also, we need 5 clean glass slides like these. Now let's proceed to perform the practical. The very first step is taking normal saline in the watch glass like this, 2 to 3 ml. Then the student proceeds to take a finger prick with the help of a lancet and spirit swab using complete aseptic precautions. For the purpose of demonstration, I will be using blood in this vial. Like this, I have to take 1 or 2 drops of blood in the saline solution. And mix it. This forms a blood saline suspension. Next, we proceed to put 4 glass slides on a clean sheet of paper under which I have written anti-A, anti-B, anti-D and control. Then we put the respective antisera on each slide like this. Next, we put a drop of the blood saline suspension on top of these drops like this without touching. and only a drop of the saline suspension on the control slide. As we know, in any scientific experiment, the control slide is not supposed to show any change. Then with the fifth glass slide, we mix these two solutions without spreading them with different edges so that in order to avoid intermixing. And we wait for 10 minutes for the antigen-antibody reaction to take place. We have waited for 10 minutes. We check for the antigen-antibody reaction by slightly rolling the slide like this. This slide does not show clumping as the solution is clear. This slide also does not show clumping as the solution is clear, and this slide shows clumping. As in this slide, the antigen-antibody reaction has taken place. It is very easy to derive the blood group from this simple procedure, as in this blood group is O positive because the blood does not have the A and B antigens. So the anti-A antibody has not reacted and the anti-B antibody has also not reacted, whereas it is O positive, that means the Rh antigen has reacted with the anti-D antibodies, and this antigen-antibody reaction has resulted in clumping. It is very easy to derive all the other blood groups from this simple procedure, and this is shown in the form of a table given at the end of this practical.
This table shows how to interpret the blood group results. It is very simple to understand. The anti-A, anti-B, anti-D and control columns show plus or minus signs, which means agglutination is present, and minus means no agglutination. For example, in the very first row, anti-A is plus, anti-B is minus, anti-D is plus, control is negative in all the slides. Therefore, we infer that the blood group is A positive. This table shows how the student has to tabulate his results after performing the practical and get his blood group from it. One of the same questions regarding this practical are: what is Landsteiner's law? Answer is, Landsteiner's law has two parts. Part 1, if a glutinogen is present on the RBCs of an individual, the corresponding agglutinin must be absent from the plasma. And part 2, if a glutinogen is absent in the RBCs, the corresponding agglutinin must be present in the plasma. Exception to the second part is absence of Rh agglutinogens from the RBCs, which are not accompanied by presence in the plasma of anti-Rh agglutinins. Second question, what is Rh incompatibility? The answer is, when Rh negative mother carries a Rh positive fetus, it can result in destruction of fetal RBCs called erythroblastosis fetalis. Third question, what is direct cross-matching? The answer is that it is the only safeguard against blood transfusion complication. It involves matching the serum of the recipient directly against RBCs of donor and again matching RBCs of recipient against serum of donor.
So today we covered determination of blood groups according to the ABO and Rh system. Next time we meet, we will be exploring the hemocytometer apparatus. Keep learning, keep growing. See you next time.